In March 2023, a mysterious disease outbreak shook Mukumu Girls High School in Kakamega County, Kenya, that led to the tragic deaths of 3 students and a teacher. As a team, Doctors Walong and Mbau and Ms. Maithya were called on to determine the cause of the outbreak of unexplained illnesses and deaths at Mukumu Girls High School.
My name is Edwin Walong. I'm a pathologist. I diagnose disease through laboratory testing, mainly in the context of the complete diagnostic autopsy and the minimally invasive autopsy.
My name is Mbau. I am a pathologist. I do autopsies, both clinical and forensic, and do lab medicine in general. That involves hematology, clinical chemistry, and microbiology.
My name is Nora Maithya. I am a medical laboratory scientist. I am also an ISO 15189 trained assessor.
So preliminary information that we had concerning the Mukumu cases was that they were potentially poisoned, and that information was circulating through the media.
In outbreaks where there is much speculation around cause of death, determining the exact is crucial. Verbal autopsy, a common practice for identifying cause of death in resource-constrained settings, lacks specificity, while complete diagnostic autopsy, the gold standard, requires extensive resources and infrastructure and is considered culturally unacceptable in some contexts. When complete diagnostic autopsy is not feasible or acceptable, minimally invasive tissue sampling, or MITS, can be used.
MITS is a technique by which we test tissues of a person who has died so that we can determine the cause of that death.
Following the deaths of these members of the school community, the Kenyan Ministry of Health sought the support of pathology experts to help determine their cause of death.
So once we heard about the outbreak, we quickly gathered the MITS team and assembled the equipment that we needed. We needed the MITS kit and also personal protective equipment. So the MITS kit contains core needle biopsies. It contains sterile containers for microbiology specimen and containers with formalin. And the PPEs that we required were gloves, disposable gowns, goggles, and masks.
The samples that were received were stool samples, CSF, tissues, and blood cultures. We had liver. We also had lungs tissue and brain tissues.
All we will do is to gently sample organs through a minimally invasive approach that leaves the body intact, and that increases the acceptability of MITS. So the families which we do engage are happy with that approach and that prospect. The main analysis that we performed were cultures to look for and isolate the bacterial pathogens that we suspected, and histology, where we take tissues through a process where we can examine them under the microscope.
We identified Salmonella typhi from the stool sample. We also identified Escherichia coli. Once we did the identification, we did our reporting, and immediately we send these results to the requesting pathologist.
MITS is a useful tool in outbreak investigation because it provides a valuable, lower resource and lower risk alternative for tissue collection.
MITS can be done in places where the complete autopsies cannot be done. MITS is even done in the community setup, and MITS is also fast to do.
Once we are on the ground, we obtain specimens within a 40-minute period, sometimes up to an hour. The main advantage of MITS is that the material submitted to the laboratory is not bulky, and the laboratory testing can be cost-effective in that we only have 2 or 3 or sometimes 5 different tissue specimens to analyze using the different techniques. And with this analysis, we can quickly arrive at a final diagnosis.
The utility of MITS is maximized when integrated into outbreak planning and response. Ideally, MITS teams should be collaborating with active surveillance teams even prior to an active outbreak.
We are very fortunate that our Ministry of Health is very well sensitized on MITS as a tool for rapid diagnostics during outbreaks. And they are already aware of the examples where this has led to a different approach to mortality surveillance. We still present cases and examples and sit in committees where we discuss the role of MITS in a wide range of surveillance activities, including outbreak responses. And in the latest Ministry of Health guidelines, we are happy that we will keep this information and this knowledge alive and transmit it to other generations and persons so that MITS can be applied in a wider context.
Established in 2017, the MITS Surveillance Alliance convinced experts to standardize MITS procedures, share best practices. Through the resource toolkit, the Alliance also provides training materials and tools to expand the implementation of pathology-based mortality surveillance.